So the Stream Pollution Trends Monitoring Program, or SPOT, is part of the Surface Water Ambient Monitoring Program. It's one of several statewide programs, and it's the only program that uses aquatic toxicology as one of its tools. And there's a lot of agencies and people we want to thank. First of all, this is all funded through the Water Board, the State Water Board, and the regional water boards. And we do a lot of collaborations with other groups like the Department of Pesticide Regulation and the Department of Toxic Substance Control and others. And we're always looking at expanding our collaborations and trying to figure out what we can do for other programs. We go to a bunch of sites every year, and we get some really great regional and statewide coverage. And if this fits into something that you're interested in terms of a monitoring program, we can talk about ways that we can leverage the efforts that we're doing so that you don't have to pay for those efforts. So I work at the Marine Pollution Studies Laboratory. It's also known as the Granite Canyon Lab, and that's our logo down there with the abalone. And it's part of the University of California, Davis, but it's out on the coast by Monterey. And I've been working at that lab doing toxicology, like Chad said, since 1992. And so I'm pretty adept at all of the nuts and bolts of toxicity testing and believe in it as a way of monitoring for environmental problems and for water quality. So let me give you a little bit of background about SPOT. It monitors the health of currently 90 watersheds statewide through measurements of sediment toxicity and contaminants. And so we go out to these watersheds, and you can see from the photos there, some of them are really beautiful and pristine, and some are kind of concrete lined. That's Bayona Creek there, which is not far from where I grew up in West Los Angeles. I used to actually play down there in that creek. I wouldn't do that anymore, knowing what I know now. But it's pretty interesting to have this wide range of land uses, both urban, agricultural, in terms of human impact land uses, and then also these open watersheds where there's very little development or agricultural land uses in the watershed. The reason I'm mentioning land uses is one of the focuses of the program is to look at trend monitoring and link it to the land use. So the things that we see in these different watersheds are very much related to the land use around the watershed, as you would expect. And I also mentioned how we have this overarching goal to use our network of sites as a framework for collaboration with other programs and agencies. I'm going to mention a few collaborations near the end of the talk that we've worked on over the last few years. But it's really about one of the original goals, one of the original causes when we put this program together back in 2008 was to use it as this backbone of sites that the regions and the state could tap into for not only the toxicology data, but contaminant data. And all of these regions can weigh in on the types of things that we're measuring from year to year. So sorry for the same set of pictures here. I don't have a lot of great spot pictures, believe it or not, even after so many years of going out there and monitoring. But why sediment? A lot of people have asked me, why would we go out and look at sediment? Why this is a water quality program? How come you're not measuring stuff that's in the water? And sediment actually integrates environmental contaminants over time. A lot of contaminants, like the really toxic stuff that we deal with, like the pyrethroid insecticides, or even the legacy contaminants like DDT and chlordane, they're hydrophobic contaminants, and they like to stick to sediments. They like to associate with sediments, and so they're transported through the watershed. And so sediment transport is a really big mechanism of moving these chemicals around the watershed. So when we go to the base of the watershed to collect these sediment samples every year, we're basically collecting everything that's been transported to that point over the last year. And it's kind of a cool way to track the trend over time. The sediment environment is also ecologically important. It's a habitat for aquatic macroinvertebrates and algal communities. And so we feel that there's a direct link between the sediment contaminants that we're measuring, the toxicology that we're measuring, and any potential impact on the benthic macroinvertebrate community. Even though SPOT doesn't do the rapid bioassessment protocols, a lot of our sites aren't really conducive to that type of protocol. But even though we don't really do that protocol, there's a lot of studies that my laboratory has performed historically that show direct linkages between the toxicity that we're measuring in the sediments and the contaminants that we're measuring in the sediments and the impact to the benthic macroinvertebrate community. When I was working on the Bay Protection and Toxic Cleanup Program back in the 90s in the marine environment, we called it the sediment quality triad. It's not something that's really been embraced as part of SWAMP, but those relationships really do exist. So sediment's a really great way of doing this tracking. That being said, we do some water column toxicity testing for the Department of Pesticide Regulation, and I'll talk about that with our collaborations. So we define watersheds, we delineate these watersheds and define them based on land use. And so this is a typical watershed that we see in Southern California. I think this one is. And you can see that rough delineation and the red dot down there, which is our sampling site at the base of the watershed. And we use the National Land Cover Database. The most recent one published is from 2016 to figure out what all of the land use is within that watershed. But the area that we're most specifically interested in is that 5-kilometer radius from our sampling site. The land use that's within that 5-kilometer radius has the most direct relationship to what we're seeing at the site itself in terms of the toxicology and the contaminants we're measuring. One thing I want to make clear now that I'm thinking about it is in some programs, people will talk about measuring chemistry in either the water column or the sediment or in various parts of the watershed. And when I'm measuring chemistry, I'm talking specifically about contaminants like metals or hydrocarbons or PBDEs or pesticides. I'm not really referring to conventional water chemistry measurements like conductivity or alkalinity or hardness. So I want to make sure everybody understands that when I mention, if I use the word chemistry instead of contaminants, I'm referring to the actual anthropogenic inputs from land use. So we roughly bin our sites into different categories, either urban, agricultural, or open lands. And you can see the definitions there in the parentheses. So I'm not going to belabor that. But we have this sort of rough binning just to kind of get an idea of representation across the state. And we do have a pretty good amount of coverage in the state. And you can see that some of our watersheds even go outside of the state and outside of the country. The Tijuana watershed down there in the southern part of the state originates in Mexico. So we have international inputs to some of our spot sites. And over the years, the sites have shifted around a little bit, but we pretty much have always had about 40, 25, 35% coverage for urban, agriculture, and open. And when we first put together the site list back in 2008, we actually reached out to all of the regions through the roundtables and asked for site lists within their regions. And so regions gave us site lists based on some of their focuses for the objectives that they wanted to figure out for their monitoring programs within each region. And so we have regions like areas in Region 5 where there's actually watersheds and subwatersheds. So you can see the darker portions of the map there show some of the overlapping watersheds that spot sites represent. Whereas down in Southern California, the watersheds are a little bit different. They're very precisely delineated. They run through particular canyons and whatnot out to the ocean. And those are more independent watersheds. But overall, we have these 90 sites representing 64 independent watersheds and 26 sub-watersheds. It's good coverage throughout the state. So what have we done over the years? This is a pretty exhaustive table of check marks for different contaminants and toxicity tests over the years. And so it's kind of separated into 3 sections. The row of numbers below the years is the number of sites we've measured each year. And we always had a target of doing 100 sites. It seemed like a good idea back at the beginning. And we started out with 92, and we immediately had a budget cut in 2009 and had to go down to 23, which was fascinating. So we were able to get that money reinstated, and we've been pretty much doing 100 sites since then. We had to cut back a little bit in 15, 16, 17 to accommodate some different parameters we wanted to measure. But since 2018, we've been at 90 sites. You can imagine that the number of sites you visit is directly related to the amount of money you have, and SPOT's budget has never changed since 2008. We have our 2008 budget, and we've just figured out more interesting ways to do more with less every year, and we're still producing some pretty good data, even with the same budget. So since the beginning, we've been doing the toxicity test with Hyalella azteca. I'll talk a little bit more about the specifics of that in a little bit, but we expose this organism to the sediments, and we essentially see what their responses are. We've been doing that organism since the very beginning. We added a second organism, a midge, Chironomus dilutus, in 2015. And the idea behind using 2 organisms is it gives you a little bit more bang for the buck. You get a little bit more data because they have differing sensitivities. And so the more organisms you can use, the more information you're going to get about toxicity. The cool thing about these 2 organisms is that they're native. They're indigenous to the creeks and rivers in California. Hyalella azteca is an amphipod. It's a little crustacean, looks kind of like a mini sand crab. And the Chironomus dilutus is a midge larva, it's a fly larva. And they're both pretty sensitive to a variety of chemicals. That variety of chemicals is the next thing here on the table. And there's a whole bunch of different chemical classes that we measure, some only in urban environments, and some in every single site. And you can see there's some blanks there on a number of years. Things like organophosphates, we stopped measuring in 2017 because we were just basically not detecting them anymore in sediments in the SPOT data set. Organochlorines, things like the DDTs and PCBs, we're going to a more periodic testing of those. We're doing those every 5 years now. And again, these are just ways of noting that we weren't really seeing big trends one way or the other, or the trends were very slow, and so it's safe to measure those less frequently. We've recently added PFAS to the list, so we have a 2021 our first data point for PFAS, and we've also measured some microcystin in sediments, and the microcystin is actually there in the sediment, just not at very toxic concentrations. So pretty wide range of stuff. So a little bit about measuring more than one using more than one organism to measure toxicity, and I use this insecticide history slide to kind of illustrate that. This is a slide that a lot of you have seen before, and I'll probably just continue showing this slide forever because I think that the information is really good, and it illustrates this pesticide treadmill or this pesticycle, and it also illustrates how the organisms can be sensitive to different things. So as I mentioned, we take these organisms, the 2 on the right there, Hyalella and Chironomus, and we add these larvae to larvae and juvenile organisms directly to the sediment in a bunch of beakers in the lab. It's pretty primitive, really. I've often called toxicity testing the blunt instrument of environmental monitoring. People often think it's more complicated than it is, but you follow a recipe, you put these organisms in the sample, and you see if they live or die, or if they grow or not. And the measurements are pretty basic. The great thing about toxicity testing, and this is the flag that I'm always flying about tox testing, is that the animals will tell you what's in the sample. You can measure all of these chemicals, but you're never going to measure all of the chemicals. And so, so the animals will tell you if the chemical is there and if the chemical is bioavailable and if it's something that can affect the animal. And so we like using toxicity testing as the sentinel organisms for this kind of thing. But with these three organisms, these are sort of our main battery of organisms that we use for looking at insecticides right now. And those arrows there sort of point to the varying sensitivities to the different compounds, which have been shifting over the years. We're not using DDT anymore, but we're still using pyrethroids quite a bit. Neonicotinoids are making a big push right now in water column testing. Fipronil is something that's used in urban environments. If you've put the anti-flea and tick drops on the back of your pet, it's probably fipronil. At least until recent years, they're introducing some new compounds now, but fipronil is the big one. And using these organisms, using multiple organisms in your toxicity tests really gives you an indication of what's going on a little bit better than just using a single organism. This figure here illustrates that a little bit more succinctly. This is some multi-species data from our SPOT program. And we started measuring toxicity with Chironomus a few years ago. And so what I did is I took all of the samples that we've tested synoptically with both species. And if you look at the pie chart there, if you were to just test these samples with the amphipod, and only look at survival, 22% of the samples would be toxic. But as you add the different endpoints, you know, the amphipod growth and then the midge survival and the midge growth, you can see that there's quite a bit of unique toxicity. So some samples obviously affect both organisms, but it's interesting to note that the amount of toxicity you observe with multiple species actually increases. And so it's pretty good to get a more complete picture by using multiple species. So I'll try to stop beating that dead horse, but you might hear me talk about it again in future presentations. Okay, so let's look at a little bit of SPOT data now, that you have a little bit of a background of where we go, what we collect, what we measure, and how toxicity testing works. This slide's a little bit ridiculous. It's kind of a silly slide, but I couldn't think of any way to really depict how there's this relationship between the toxicity we observe, particularly with amphipods, the pyrethroid pesticides that we measure in the environment, and the urban land use, particularly this kind of residential land use. And it's this three-way relationship that I'll kind of talk about through a series of different figures. And this is sort of the main story that SPOT's been telling for the first 10 years of data. We came out with a 10-year report. I think it came out formally last year, and we analyzed data from 2008 to 2017. So the data I'm going to be talking about in the next few slides covers those years. So let's first talk about pyrethroids. This is the pyrethroid leg of the triangle. So on the left side of the table there are a list of what we would call active ingredients, specific pyrethroid compounds that you would find in various off-the-shelf products or agricultural products. It's an interesting thing. A lot of people think that pyrethroids, being pesticides, would be seen most predominantly in agricultural watersheds, or watersheds that are influenced by agricultural runoff. But we really see a lot of these pesticides in urban areas because essentially people don't like bugs. They don't like ants and cockroaches, and you can go down to Home Depot and you can pick up one of these products that contains one of these chemicals, and you can spray to your heart's content around your house, And then if you're in the know, like us water quality experts, you might wait a few days before you water your lawn. But people like my dad, he just waters his lawn right after he sprays for ants. And so even with folks that that are related to water quality experts like myself, it's difficult to convey how to behave properly when you're spraying pesticides. So we have a lot of this kind of behavior in urban environments. And so you see a lot of detections of these chemicals in urban environments. And if you look at the number of detections, which are the sort of right portion of the table there, there's pretty significant increases really of a number of pyrethroid insecticides in all of the land use areas. And the biggest increases we see are in the urban areas, of course. And then the trends in the concentrations, which are sort of on the left side, the most significant increasing trends are in the urban areas. So we know how much pyrethroid is getting sold in the stores. We know how much the growers use in the agricultural environments. And then we can see these increases in both use as well as detections. So now I want to show you the chemistry again, but expressed as the potential to cause toxicity. And so this is a little bit of a tricky concept, but we use it as a way of kind of simplifying all of the thousands of pyrethroid concentrations we're looking at into its potential for causing toxicity. We can convert these pyrethroid concentrations into toxic units. And if you take a particular threshold concentration, so the amount of pyrethroid, an individual pyrethroid that's going to cause toxicity, if you have a threshold like a median lethal concentration or an LC50, you can divide that number into the concentration that you've measured in the environment. You can create this toxic unit number. So ideally, if the relationships hold true, one toxic unit of a single one of these chemicals would cause about 50% mortality to the organisms that are exposed to it. I mean, we have some sites that have, 20 or 30 toxic units. You have these huge amounts of these chemicals in some of our sites. But this is the numbers of toxic units plotted over time in a bar and whisker plot. And it's essentially giving us an indication of the trend of toxic units over time. And it's a very subtle increase, but it is a significant increasing trend in median numbers of toxic units. And so we are seeing the potential to cause toxicity by pyrethroids increasing over time. I'm sure I'll have some questions about that at the end. That's a bit of a hard concept to grasp, but just keep thinking about the potential to cause toxicity. So what makes up all of these toxic units? One of the things that we're interested in is trying to ascribe some causality to what we're finding. One of the great things about toxicity testing in the laboratory environment is you can do a number of sort of diagnostic things to figure out if the pyrethroids are actually causing the toxicity. And so we're very interested in what's making up these toxic units. And believe me, the pesticide industry is interested in what's making up these toxic units as well. And you can see from this figure, over the years, bifenthrin really drives the number of toxic units we're observing in these samples. And bifenthrin is one of the most long-lived of the pyrethroid insecticides. They have half-lives, in months to years. And it's also one of the most toxic and widely used. And so that's really, it's kind of obvious when you think about it, but bifenthrin really drives the numbers of toxic units we're seeing. Okay, let's talk a little bit about the toxicity leg specifically of the triangle. So we've talked about the pyrethroid leg of it, and this is a little bit of information about the toxicity side of it. So as I mentioned, we're very interested in not only measuring whether or not a sample is toxic, but we want to know what's contributing to that toxicity that we're observing. And I also mentioned that we can do these diagnostic tests to try to figure out if pyrethroids are actually contributing to that toxicity. And one of the tests we do is we conduct our amphipod toxicity test at a colder temperature. The thing about amphipods and a number of other organisms is the way pyrethroids work. The amphipod metabolizes the pyrethroid, and at a warm temperature, it can metabolize more of that chemical and sort of detoxify it. And at the cold temperature, their metabolism slows down and they're unable to process the chemical, and therefore the chemical actually becomes more toxic at a colder temperature. And if you think about it, across the state, 23 degrees, which is the standard temperature for toxicity tests, for these toxicity tests, the water is not really 23 degrees across the state. It's actually closer to 15 degrees. And so we've done some tests at 15 degrees and 23 degrees side by side. And so every dot that you see in that left-hand figure has a corresponding dot in the right figure. And you can see how at a colder temperature, basically the take-home message here is there's way more red dots on the right. You know, we have much more toxicity when we do these tests at colder temperature. And if you look at the toxic units, which are across the x-axes of both of these figures, you can see a shift in that toxic unit threshold. You know, you get a lot of toxicity somewhere between 3.5 and 4 TUs there on the left, but it's a little bit shifted more down to 2 in the figure on the right. And so this is a bit of a diagnostic. It indicates that, you know, the pyrethroids are definitely contributing to the toxicity we're observing in the lab tests. Now, I haven't mentioned land use yet. This is the third leg of the triangle, urban land use. And so this sort of drives the point home about where the toxicity is happening and where the pyrethroids are being observed. And so in both of these figures, on the x-axis, you have percentage of urban land use in 25% increments, with the most urban sites being on the right-hand side. And then the left figure is the percent survival in our test organisms. And in that greater than 75% urban land use, you can see that there's a much bigger bar there. We have much lower percent survival on average when we're testing urban sites. And then, of course, you know, the far right bars in the right-hand figure show much higher total pyrethroids in nanograms per gram. So these are actually, you know, mass measurements of pyrethroids. The highest concentrations are in those urban areas. So we have this pretty clear relationship among these three legs of the triangle. That's the main story for SPOT for the 2008 to 2017 data set. So pyrethroids aren't the only thing we're measuring, of course. You know, I showed you that table that had a big list of things. And so I'm not going to go through each figure one by one, but I do want to just give a little bit of a summary of some of these other results. And I'll show you the fipronil concentrations just so we have something on the screen while we're talking. Fipronil is an urban insecticide. I mentioned this is the one that's used a lot in flea and tick applications for pets. And we started measuring this back in 2013, knowing that it was a contaminant of emerging concern, and we wanted to kind of get a jump on potential trends. And fipronil is a parent compound, which is the figure on the left, and it has some breakdown products, fipronil sulfide and fipronil sulfone. And we're not really seeing any significant trends, but we are measuring it in all of our urban sites. We are seeing it at a number of urban sites. And in some cases, it is just barely approaching toxic concentrations, or concentrations that we would consider as potentially contributing to the observed toxicity. There's a couple of other urban things that we measure, and it's PBDEs and PAHs. And PAHs are hydrocarbons. You know, it's part of having fossil fuels in our world. We're not really seeing any trends in PAHs. PBDEs are flame retardant chemicals, among other things. and we are seeing some increases in PBDEs. We've been doing these legacy contaminants like DDT and PCBs, and we're seeing either downward trends, slight downward trends, or stable trends with those chemicals. And then we've also been measuring metals, and we focus on the metals that are that are most likely to contribute to the toxicity we're observing, like copper and zinc. And we're not really seeing any trends with copper now in any of the land use areas. We're expecting to see some downward trends in copper over time because of the phase out of copper brake pads. And most interestingly, we're seeing concentrations of zinc increasing statewide. We haven't dug into that too much to really figure out why that's happening. So now I'm going to segue a little bit into our collaborations and discuss some of the things we've been working on with DPR and with DTSC. And one of the things that we've had a relationship with DPR, our lab, for a number of years. We've done a lot of agricultural treatment stuff and agricultural runoff projects with DPR. So we have a member of the DPR staff on our scientific review committee. So it's pretty cool to be able to do stuff with DPR and kind of have this cross-agency relationship. I mean, the water board and DPR, you guys are in the same building, so you know it's good to be working together on stuff like this. But in 2012, DPR changed some labels for their for pesticides in California for commercial applicators. There's a photo here of I'm not sure if this photo is staged or not, to be honest. Because it's a commercial applicator, what appears to be a commercial applicator without the proper safety gear on, spraying pesticides on the side of a house when it's clearly been raining and might be raining again. So basically doing a lot of things perhaps incorrectly. And DPR came up with some label changes that eliminated some of these practices. Instead of the sort of broadcast spraying some of these professional applicators used to do, they have to spray a very tight bead of insecticide at the base of the wall as opposed to up the side of the wall like they used to do. And so one of the things that DPR was interested in is trying to track whether or not these label changes were having an effect in the receiving systems that we sampled. And so we took a couple of DPR sites and a couple of spot sites and started sampling them more frequently and tried to get a little bit more resolution of whether or not we were having changes in the toxicity, which is the Hyla azteca column there, and changes in pyrethroids and fipronil. And the good news is that there were actually some significant decreases in these two chemical classes based on the sum concentrations as well as the sum of the toxic units that we were measuring. So we are seeing some significant decreases. We can only assume that that's related to the label changes, which is good news for everybody. Interestingly enough, though, we are seeing some slight increases in the toxicity to the growth endpoint, but those are probably inconsequential compared to the significant decreases in concentrations of those chemicals. Another collaboration we've been working on with DPR is we've been providing some water column toxicity testing to accompany their surface water monitoring program. DPR goes to some focused sites in both agricultural and urban areas in a couple of spots in the state to look at the chemical concentrations, and they do this big suite of chemicals, insecticides, insecticides, herbicides, and fungicides, and the CDFA laboratory does these analyses. And they do a little bit of toxicity testing with some of these samples, but they haven't had a really big amount of coverage on these samples from year to year. And so for the last couple of years, we've been trying to provide a little bit more coverage with their surface water samples to give them a little bit more data. And there's, you know, as you would imagine, in some of these sites, there's a lot of toxicity. There's a lot of dead organisms and impacted organisms. And in some cases, you can see direct relationships to the concentrations of these insecticides that they're measuring. And DPR is just now starting to really tease apart that data set. I mentioned the data briefly in our 10-year report, but that's just the tip of the iceberg of the data that we've been collecting. So there's a lot more data to be processed. And I know a couple of folks over at DPR that are working on that. We also recently had some extra funding in the statewide chemistry contract, and we reached out to DTSC to see if there was anything they were interested in measuring at spot sites. And so in 2019, we were able to collect some extra samples at spot sites and measure these organosilicon products, compounds that are in personal care products. I won't bother pronouncing them, as well as galaxolide, which is a synthetic musk used in fragrance. And these were chemicals that were kind of on the watch lists at DTSC, but they didn't really have much in the way of baseline data for statewide. And so we were able to do this, like, statewide shotgun blast of sampling and get DTSC numbers for these compounds. And so they're in the process of sort of going through those data sets right now and trying to figure out what it means and what we should focus on next. And so it's kind of cool to have these compounds kind of come to the fore. They're contaminants of emerging concern. DTSC is interested in them, and it's great that SPOT can provide the infrastructure to collect the samples and do the analysis. So what has SPOT reporting looked like? We started this program in 2008, and we came out with a report with 1 year of data. So it was a trends program with 1 year of data, and we wrote a report, which is just like us to do. And it didn't say much in terms of trends, obviously, but it did give an idea of the concentrations and the significance of the things we were starting to measure. Since then, we've tried to come out with a report every couple of years and kind of continue to update the trends. And after 5 years of data, Katie Siegler was able to produce a paper. And so we published a paper in Environmental Pollution on our first 5 years of data. And we continued with our 7-year report. And then most recently, we decided to just do a 10-year report. And we're in the process of preparing a manuscript for that report as well. So we'll have 10 years of SPOT data. What we're really interested in is the future of SPOT reporting. These reports take a lot of effort to produce. They're kind of onerous to read, and people pick what they need to out of them. But we feel like the SPOT reporting system, if you will, can become a little bit more nimble. And one of the ways we want to do this is through these toxicity and chemistry dashboards that the State Water Board is putting together. I know, Michelle, I think I saw you on the list of folks that were joining the call earlier, and Michelle Tang has done just an incredible job of building these sort of interactive dashboards that tap into the databases to produce real-time trend analysis of what's going on, not only with SPOT, but with other programs as well. And we think that this is sort of the future of reporting. We'll be able to pick out these individual trends or individual stories. And if regional boards are interested in kind of having things interpreted or knowing more about what's going on, or not just regional boards, anybody really, any agency group, we can help interpret that and create sort of small, more focused write-ups on the different stories that are happening throughout the state. And I want to apologize to Michelle because I'm using like an old screenshot version of the dashboard. It looks way cooler than this now, and I'm really excited about seeing it come out. So lastly here, what is the future of SPOT? And we've talked about this kind of, blunt instrument toxicity testing, this blunt instrument way of monitoring environmental impact. We've talked about the contaminant monitoring, and we've talked about the relationships and the stories we found with these pyrethroid insecticides. Talked about how we can report these things to people and how we can get the stories out to people as quickly as possible. What are we going to do in the future? Now, historically, the monitoring programs I've worked on have all kind of focused on these legacy contaminants, things like DDT and PCBs and toxaphene, and we know that the success of these monitoring programs that look at contaminants is really limited by the analyte list. Even though we want to keep measuring those legacy contaminants, those historically used contaminants, and monitor their trends, there's so many more things we need to be measuring. And the things that we need to measure, I mean, all of that's limited by funding. If we had unlimited amounts of money, we could measure everything. And so the big question that we're always asking is, what do we measure next, and how often do we need to measure it? And so that's our big question, and how we decide to spend the money every year. And we want to expand our connections to existing CEC efforts. Like, we know SFEI and OPC have contaminant emerging concern groups. And so we're keeping an eye on what those groups are coming up with in terms of analyte lists, and we want to try to focus on those lists for targeted analysis. And an example of that is PFAS. We're measuring that this year. Our scientific review committee agreed that was a good thing to start getting some trend data on. But we want to know what the next thing is going to be. And that's for the targeted analysis. We're also wanting to do some non-targeted analysis. And so we have a pilot project that's just getting underway. We've been severely delayed with COVID, among other things, COVID, visa issues, and injuries, believe it or not. But we're finally getting this project underway to do some non-targeted analysis at a subset of SPOT sites, and couple that non-targeted analysis with measurements of sublethal behavioral endpoints in our toxicity test. So not only are we looking at a different way of analyzing things that we don't normally analyze, but we're looking at some different toxicity endpoints. We know these are kind of novel approaches right now, particularly the toxicity approach, but we feel like someday these approaches will be incorporated into the regulatory framework and get used a little bit more routinely. There's a number of researchers up at Davis that are looking at ways of incorporating things like adverse outcome pathways and bioanalytical tools and other types of measurements that hopefully will be less expensive and will have higher throughput capability that can be incorporated into the regulatory framework in the future.